Good afternoon, everyone. Thank you so much for coming today. I know it's not easy. Very much appreciate your attendance today. We only have about 10 degrees now. And of course, I want to appreciate our speaker, Dr. Chase Weidmann, from the Department of Biological Chemistry in University of Michigan, our local colleague. Um, uh, Chase has been working on the RNA biology for long times. Let's make a very brief introduction to his background. He got the bachelor degree of biochemistry in University of Rochester, and later he received the doctorate trainings in University of Michigan's here in the Department of Biological Chemistry, and received the PhD degree in 2015. And after that, he spent a couple of years in University of North Carolina, right, to finish the first round of postdoc training, and spend a very short period in St. Louis, in Washington University in St. Louis, to learn the skill set of the proteomics. However, Dr. Weidmann spent a lot of time in RNA biology from his PhD training until now, and he works a lot in the RNA-protein interaction. So I think his talk today focus on the RNA-protein interaction network, right, in different types of disease and cancer. And I want to highlight that Tracy is a very successful trainee, faculty, and mentor. He receives the training grant from the PhD periods and the postdoc training periods. And also, recently he received nominations of the Outstanding Faculty Mentor Awards in University of Michigan. So overall, I think that's a very successful and amazing career path. So I look forward to the talk today. Thank you.
Thanks for that introduction. Thanks everybody who is here in person. I know it was probably hard to get here. Looking forward to some days where we're only at about freezing coming, just better than today. So yeah, I'm Chase Weidmann. I'm here in the biological chemistry department at the University of Michigan. And my laboratory studies just very broadly RNA in biology and disease. And I always have to start by a central dogma slide. When most people are thinking about RNA, they're thinking about messenger RNAs that code for protein. And I always like to point out that in reality, protein coding sequences are only a really small fraction of the human genome. I'm actually being pretty generous. I think some estimates put it at less than 1% of sequences. And actually, even those sequences themselves, right, mRNAs start with a lot of non-coding sequence attached to them in the form of introns that are important for their processing, but are ultimately spliced out. And all told, probably about 80% of the human genome is transcribed in any one cell, and those sequences, by and large, do not encode protein. That means that about half of the genome, right, is transcribed into independently acting, non-coding RNAs. And that's including about as many unique genes as there are unique protein coding genes. That's not so surprising, right? RNA can fold into complex structures just like proteins can. So it's not, again, not surprising that there are lots of functional RNAs that do a lot by themselves. But as it turns out too, if we look at the things that are made into proteins, about 10% of the human proteome actually interacts with RNA. And these RNA protein complexes have a bunch of very important diverse functions. Think of telomerase, the signal recognition particle, the ribosome, RNase P. These are all non-coding RNA protein complexes that are ultimately involved in every critical step of gene regulation. And this is only a small subset of the ones that we actually understand. There's a lot more unexplored, unknown non-coding RNAs that we don't know what they're doing. And if we go even bigger, even larger RNA protein complexes can even assemble into these distinct non-membrane bound compartments inside of a cell. And these happens when RNA and proteins are able to make RNA-RNA, RNA-protein, and protein-protein interactions. These multivalent interactions enable them to sort of create boundaries within a cell that include or exclude specific molecules. And the last feature of RNA protein complexes I'll talk about is the fact that they're much larger than your average protein or protein-only complex. So these are huge molecular machines that do a variety of complex cellular tasks. And that's just, again, the small fraction that we understand at this point. The one thing I like to think about this is thinking about their size, just physically, the amount of unexplored surface area that these things are presenting. It's interesting to me as potentially new ways to target things therapeutically. Okay, so ultimately, all the projects in my lab are focused on how RNA and proteins come together to do things and how these interaction networks are disrupted and how that results in diseases. So the more we understand about the assembly of these complexes, the better chance we'll have to actually manipulate or target them. So how do we actually ask questions about these complexes in the lab? And the way I like to say that we do this is literally, because this is what we do. We just dump chemicals onto cells and see what happens to the RNA. We use chemical probes that are specific to certain RNA features that allow us to measure different events. And then once we have an RNA that has been modified, we can convert those chemical adducts into readable information. So through this process called mutational profiling, or MAP. In MAP, there's specific buffer conditions we use that will cause certain reverse transcriptase enzymes to leave behind gaps or non-templated nucleotides when they encounter these adducts. So at that position in the product cDNA, right where there was an adduct, you'll see what ultimately can be read out as a mutation just by sequencing it. And so once those mutations are there, right, you sequence it deep enough, you can measure the frequency with which these mutations occur. And mutation frequency is proportional to the reactivity of a nucleotide to whatever chemical probe you're using. Meaning we're going to get nucleotide resolution chemical information for any RNA that we're interested in. And so we have lots of different chemical probes that reveal different attributes of RNAs. And it's kind of surprising that this method is sort of robust enough to accommodate lots of different kinds of adducts. So if you can put something weird onto an RNA nucleotide, you can get a reverse transcriptase to see that as something weird and then do something funky in the right conditions. So first, I'm going to describe a couple stories using chemical probing to define how certain RNA structures mediate biology and disease. A larger portion of the talk is actually going to be how I'm thinking about developing an approach for mapping these protein networks on RNA in transcriptomes. And then I'll have a very short foray at the end into using map technology to screen for interactions between RNA and small molecules. So as a way to actually do drug screening on RNAs inside cells. Okay, so I'm going to start with an example of the most established map approach, which are using these shape reagents that rely on some kind of modification. Shape reagents rely on 2' hydroxyl acylation to report on conformational flexibility of nucleotides. You can do this on RNA in vitro or in cells. And you use this information to model secondary structures with experimentally derived data. And first, I'm going to do a detour. I literally added this morning thinking about this audience. So artificial intelligence has revolutionized a lot of our world recently, including aiding in the modeling of three-dimensional protein structures. So here's an accounting from this morning that the number of computed structure models, mainly from AlphaFold that's in the protein data bank. And you might think that modeling the 3D structure of RNA is right around the corner, and maybe there's plenty of people who do think that that is going to happen, but I'm a little skeptical at this point because AlphaFold ultimately benefits from a dramatically larger existing data set of protein structures, right? And probably half of this data set has been generated maybe in the past decade, and we're lagging behind on the amount of information to be able to train a model that will be able to predict accurately the 3D structure of RNAs and especially RNA protein complexes. So while AI is advancing, we're probably a few years away from doing this same kind of trick with RNA. But what we do have right now for sure, and we've had for a long time, is a pretty robust set of empirically derived thermodynamic parameters to describe the free energy of RNA secondary structures. So how RNAs can base pair internally. And as a reminder, RNA can assume multiple states, right, by taking on different conformations of base pairing. Some of those structures are going to be very similar in energy and likely will populate an ensemble at similar rates. There's math. Some of you might be better at math than I am, but we have equations and dynamic programming algorithms that can model which secondary structure will dominate when there are multiple conformations available based on these computed free energies. From a set of highly probable structures, you can calculate the probability of any base pair in an ensemble of RNA structures. I bring this specifically up because I have some data that is going to be what you generate from this. Okay. So using, I say existing algorithms, but I realize this slide might be old from a paper in 2013. Maybe there's always better incremental steps being made at predicting from de novo RNA structures. But in general, you can get about 70, 80% right the time without doing any sort of experiment. But if you use shape chemical probes and do an experiment and ask, what does this RNA actually look like in the system that you're testing, you can boost that accuracy to closer to 90%. So how do we do this? Well, I told you already how we do it. We dump chemicals on cells, but I'll go into more detail on how you might use a chemical probe to measure the secondary structure of an RNA. So this is an example I always show. This is the U1 small nuclear RNA whose structure is known. And I'm representing the structure in two ways here. On the left, in this linear fashion, where these arcs are representing base pairs. And then on the right is, maybe you've seen more of these like airport-looking diagrams of what an RNA secondary structure looks like. This is just to orient you to how I'm going to be plotting the data. So if I extract RNA from cells and probe with a shape reagent, 5-NIA, you'll see a reactivity pattern that looks like this. This is a good reactivity pattern, what you would expect based on the structure of this RNA. You can see that base paired stems are lowly reactive. This is these bars in black, same thing plotted on here. And then the loops and single-stranded regions, right, are more reactive, plotted in red and yellow. And remember, I talked about being able to predict the probability of any single base pair being formed. That's what this arc plot is representing here. So these represent the computed probability for each base pair. And in this case, one structure is highly favored by the experimental data. You can see all these arcs are green. That means that these base pairs are represented in greater than 80% of those low energy structures. I missed something to say. Oh, yeah. Also, these arcs are actually generated from the shape data, right? You can augment the free energy calculations by using the shape model as a restraint. Okay. This actually does a really good job. So it's identifying all of the known base pairing structures in this. And in addition, it's also predicting this extra helix, although that kind of makes sense. Inside a cell, this region is actually bound by a protein complex. I'll talk a little bit about that later as well. So when we probe this structure not in cells or with proteins, it will be predicting this helix as well. Which might be true, but irrelevant inside the cell. So in my postdoc, I used a bunch of these chemical probes to evaluate a number of different long non-coding RNA structures in a bunch of different systems. And around that time, I met another postdoc at Dana-Farber named, her name's Karen Schmidt. She had recently published work on a long non-coding RNA called Slinker-1 that was tied to lower survivability of melanomas. So ultimately, in more aggressive melanomas, you get overexpression of this Slinker-1 long non-coding RNA, which led to a higher prevalence of metastatic disease. And ultimately, she saw that it depended on this BRN3A protein. It's a transcription factor along with the androgen receptor. And together, these activate expression of things that promote metastasis, like matrix metalloproteinases. And they sort of wanted to understand a little bit more about how this RNA is working. And I told her that if you can narrow down the part of the sequence to what's actually necessary, small enough region, it would be definitely straightforward to assess with shape. And that's exactly what she did. So here's a experiment where a cell line is being presented an extracellular matrix and giving the opportunity to cross over, but only the right expression profile in a cell enables it to do something like that. And invasion across this barrier is a proxy for measuring metastatic activity of a cell line. So indeed, when you express this long non-coding RNA, you see that cells invade across this barrier. And deletion of a small conserved region could prevent that invasion activity. Further, if you just take out that small chunk by itself, if you express that, it's responsible for pretty much all of that invasion activity from the Slinker1 RNA. So, knowing that, could we find an RNA structural motif based on experimental data that is likely responsible for uniting these transcription complexes together? So I took patient-derived melanoma cells and extracted RNA for shape probing. And this was, at the time, not a very exciting result because you spend enough time looking at all the control RNAs and they all look like this. And then you do it on something cool and it's looks like this, which really, if you just look at the data, right, it's pretty broadly reactive to the shape reagent across this region. So this was probed in the context of the whole RNA. This was in patient-derived cells. We're just amplifying just this region to look at it. So what this really means is that it's probably unlikely, just looking at the shape profile, unlikely to be forming significant, stable secondary structures. So I put the U1 RNA up here from the same cell line, which you can tell has a single stable structure just by looking at it because it has this sort of characteristic pattern of extended regions of low reactivity and then hyperreactivity at loops. So if you gazed at this sequence, maybe there's potentially a structure that might be stably forming right there. So if you model base pairing, this yields a bunch of competing, similar free energy, low probability pairings. And so even if this region is forming a structure, it's probably sampling a bunch of different multiple conformations. So maybe this is inherent to the system. Maybe it's the flexible landing pad for proteins to bind to. And thinking about protein binding, indeed, the sort of more flexible region actually overlaps exactly this part that was necessary for invasion activity. And ultimately, this is responsible for binding to that transcription factor, BRN3A. And now I mentioned before that this region that they identified, right, was from conservation. So it has homology to another non-coding RNA that binds to the androgen receptor. And this is protein that guides regulations or binding to DNA in a number of different ways. And by EMSA and SPR, we found that the androgen receptor's N-terminus is actually capable of binding to these pyrimidine-rich motifs. And it was interesting that it was the N-terminus that was binding. It's not the nucleic acid binding domain that was known to be there. It's binding in this N-terminal region, which sort of leaves the DNA binding region available to target different genes. So 2 of these motifs exist in this region that's expressed by itself as causing this invasion phenotype. So meaning that Slinker1 essentially has the capacity to join together the targeting activity of the androgen receptor with the transcriptional regulatory activity of BRN3A. You could actually use shape probing inside cells to figure out if there's protein binding activity. So if you take an RNA outside of a cell and remove all the proteins, you can get a shape profile that shows its intrinsic structure. But if you probe directly inside cells, you can look for changes in that reactivity profile that might be indicative of other interactions happening inside the cell. And indeed, we do see a bunch of significant protections in this region, including the most highly protected region being in this BRN3A binding region. And we saw protections at both androgen receptor motifs as well. So the idea is that this flexible region within this highly expressed and aggressive melanoma long non-coding RNA is bringing the androgen receptor and its targets together with this BRN3A binding site, forming a non-canonical transcription complex that ultimately is now promoting metastatic gene programs. Also from our SPR data, we knew that the receptor couldn't bind to the androgen receptor motifs if they were base paired, i.e. if they were in sort of a helix, the androgen receptor wouldn't bind to them. So the flexibility of that region was sort of necessary for this activity to occur. And you could also leverage that to think about targeting it therapeutically, because you could inhibit this metastatic activity if you use oligos that are complementary to those androgen receptor binding sites. So if you sort of zip that up into a structure, it no longer will recruit this activity. And similarly, if you just soak the cells up with just a small chunk that can occupy that N-terminal binding site on the androgen receptor, you can also inhibit metastatic activity. And so I'll stress here, and Matt, I'm getting to the thing I talked about earlier, that, right, it was now you can make the argument that the absence of a complex structure is still a structure. A single-stranded region of an RNA can be important, but the absence of some kind of complex-looking structure in RNA is what actually enabled this phenotype. But not all long non-coding RNAs behave this way. So this is, for comparison, this is the monk long non-coding RNA, which controls differentiation during myogenesis. So you can see here already that this looks a lot more like this U1 profile. And indeed, monk RNA appears to fold into multiple distinct stable structures. And most of these regions also show changes in reactivity if you probe them in cells, suggesting that there's widespread interactions with proteins inside the cell as well. So my collaborator at the time, Ruja, had a number of established assays in mouse myoblasts that measured promyogenic function and was able to test how deletion or mutation of each of these different identified structural motifs affected the phenotypic outcomes. And what was cool about this is that literally you couldn't touch any part of the structure of the RNA without screwing up its activity. So essentially, full activation of one of its targets, the MyoD1 gene, required pretty much every motif to be intact, especially dependent on this CH1 and CH4 motif. And what was still really interesting is that not all of its gene targets required all of the structures in the RNA. Some of them only required some of them, and including a major target that doesn't even require the one that most of the other targets required. Sorry, I'm saying confusing things. Hopefully you'll understand. But MyoG didn't even require the CH4 motif. Further, you don't see the mutant complexes able to complement one another. If the idea is like you need a presence of a certain RNA structure to recruit a specific protein to a specific location, you might think just combining one that has a mutation in one of the structures and one that has a mutation in another, putting them both in at the same time while you're recruiting them to the same place, you should be able to recruit the same proteins. But alas, this could not rescue the effect, meaning that these RNA structures have to actually work together on the same molecule of RNA in order to do what they're doing, except for the targets that didn't require both of them. Okay. So the point of that was to show that by structure probing, you can definitely identify important functional motifs in RNAs, but you won't know whether a flexible region or a complex structured region are more important. So Slinker and Monk are sort of opposite ends of the spectrum here. You got a short, unpaired flexible region that bridges a complex together. And then in Monk, you need all of these different complex motifs to come together to regulate specific targets. And depending on which target it is, you need different combinations of them. But what was true in both cases is that they needed to bind proteins to do what they're doing. So if you want to find motifs in RNAs that you are just starting to study, and you want to find the motifs that matter for function of those RNAs, if RNA structure isn't always a guarantee of importance, my recommendation would be to just directly look for protein binding sites on RNA. So now I'll show you a map approach for protein footprinting that I developed and am continuing to develop. This involves, at its base, a different chemical probe. This is SDA. This has a amine reactive group here and a photoactivatable diazerine. So the amine reactive group selectively reacts with flexible free amines like those in lysines. Lysines are one of the most prevalent side chains in RNA binding domains, along with arginine. So upon photoactivation of this with long wavelength UV, the diacerine group will rapidly cross-link these labeled lysines to nucleotides in close proximity. So this will be essentially in the sub-20 angstrom range. Unlike other methods of RNA cross-linking, this is going to be largely agnostic to the identity of the nucleotide base. A lot of existing approaches are very specific for uracil. So once you make these cross-links, you digest away most of the protein and you leave behind short peptide adducts. And these adducts actually serve great as map substrates. We can use mutation frequencies here as a measure of protein binding on an RNA. And because we are actually reading through these adducts, sometimes you can get the observation of multiple protein binding events on the same RNA molecule. And in the sequencing data, you essentially look for correlations between mutations, and those represent protein-protein communication on RNAs, meaning that we have a way of not just footprinting where there's protein binding sites, but finding where there is networked protein complexes on RNAs. And so what does this information look like? You're going to see U1 a lot. We used U1 again as sort of a control because we know what the structure of this RNP looks like. And so here now on the left, I have 5' to 3' just where the binding sites are for proteins. And then I've got this 2D, 3D representation on the right. And so I wouldn't be talking about this if it didn't work, but yeah, nucleotides with high SDA reactivity correspond to known binding sites really well. And if you zoom in and look at where those cross-links are happening, they're happening at lysines, where you would expect the drug to be acting. Not only that, the proximity to a lysine was correlated to how high the reactivity was. So that was refreshing. And we also capture this correlation information. So this is a highly stable complex. It doesn't work unless it has all of these things here together to bind to splice sites. And indeed, you would expect all of these protein binding events to be correlated in some way. This is an organized assembly process. So to test if I could use this RNP map approach to identify functional motifs in new RNAs, I probed the exist RNA. This is a 20,000 nucleotide long non-coding RNA responsible for X activation. This is where X chromosomes are silenced in cells that have more than one X chromosome. And exist actually moves along the entire X chromosome to maintain an silencing compartment. So you can see this granule here. And loss of this compartment is seen in a number of gynecological cancers. So just looking at exist, you see that it has these tandem repeat elements, a number of different ones. And at the time when I was studying it, what each of these elements did wasn't super clear. So you did RNP mapping. I did it. So we did RNP mapping of the exist RNA and found this E region was the site of most protein binding as well as the most highly networked region in the whole RNA. And we thought, well, that probably means that this is important. We looked at, because there was existing eClip data on some of the RNA binding proteins that recognize this region. And when we did a combination knockdown of some of these proteins, you start to see loss of this RNP granule compartment of exist inside cells. And what was interesting about this is if then we took that E region, right, and just stuck it in a random other RNA, so this is a mRNA reporter, something totally different, you could still get it to form these RNA protein granules, except now they're in the cytoplasm. So RNP-map was sort of able to find this super highly networked region that ultimately seems to be a granule-forming prone region of RNA. Ultimately, the point is that RNP-map seems to be a good tool in pinpointing functional regions of RNAs. And since starting my independent laboratory, I've been continuing to try and improve the RNP-map method. And one of the first things I did was just go and find the funniest sounding acronym thing and try and put it in my experiments. No, there was an important reason to use this organic phase separation method because it was a way to enrich for cross-linked RNPs, and since that's a major part of my process, I thought that would help. Essentially, cross-link protein complexes end up trapped in this interphase layer, which we can then purify. And indeed, this did great in improving the signal to noise for the detection of protein-binding nucleotides. And more importantly, right, so it also enriches for multiply modified reads, meaning RNAs with more than one modification per molecule. So in the original version, you can't even really tell that there's a difference between these two distributions. There's lots of background correlations that you have to filter out and would skew the data or dampen how sensitive we could be with finding the actual networks. But when you do this OOPS approach, you get clear separation and also nice to our wallets. You could sequence way less and still get as complex, if not more complex, information-rich correlation networks. Okay. So now I'm going to maybe embarrass myself a little bit. So I have no formal training in statistics or bioinformatics. So the machine learning I'm going to apply is going to be potentially very rudimentary. But because the signal was increased, we thought we could actually be more confident in assigning where protein binding sites were. So before we were using empirically defined thresholds from known RNP complexes, of which there's, again, if you remember, there's only really a few complete RNA protein complex structures solved at atomic resolution. But our increased signal really enables us to be more confident without relying on those potentially biased data sets. So now we use a random sample consensus approach to ultimately find a background model within each new data set and then identify outliers, which we can then assign confidence levels to, depending on the strength of the model that comes out of it. Because there is still some bias, way less than other existing methods for doing these cross-linking experiments, but there is still some do this analysis for each nucleotide independently. And does this actually help? And the answer is yes. So the new analysis, and especially this phase separation step, really enhances our ability to find nucleotides that are in close proximity to lysines. So the significant signal here is most accurate at detecting nucleotides that are within 15 angstroms or less of a lysine. So I'm still working on improving the method, but ultimately the goal is to map these kinds of protein network interactions across transcriptomes so that we can actually pull out binding profiles that target groups of RNA that we can then assign to different proteins. Think about simultaneously trying to identify all of the RNAs that are bound by proteins and what proteins are binding them, and at nucleotide resolution where those binding events are happening. That would be the dream. So we do have known binding sites from eClip data, along with a variety of binding site predictors. So we'll likely be able to assign profiles that we generate to specific complexes if we do RNP-map in an eClip cell line to start with. Then we could use the resulting profiles, right, to map binding sites in other cell lines where we don't have eClip data. And I expect we'll also discover a whole bunch of profiles for RNA binding proteins that we don't have any information on, because I think only maybe 200 RNA binding proteins out of thousands that exist are actually in eClip data right now. So there'll be a big opportunity for discovery of new kinds of complexes, likely by mass spec methods. But before going too deep into, I'd love to say I have that working and we can already predict those things, but my sense is before we go and try a big experiment, I just want to make sure that it's probably going to work. And so there's just a couple questions I want answered before we move towards this transcriptome-wide profile generating approach. And that is, how sensitive are RNP-map profiles within their sort of interaction environment? And would the same complex, i.e. set of RNA binding proteins, even result in a similar profile? Can we even do matching? Is it worth trying to pull that out of a big data set? I think these are important questions to ask before we can think about deciphering these things transcriptome-wide. So we've been applying RNP-map to a number of different projects. One is this polymerase RNP. This maintains the ends of eukaryotic genomes. And so one part of the complex is pictured here. this TCAB1 protein is responsible for localizing it to a specific RNP granule in the cell. So lots of non-coding RNAs bind to this discarin protein. This one binds to. That will end up in multiple nuclear RNP granules. But this TCAB1 protein exclusively localizes to one type of RNP granule, the Cajal body. And that makes it so that telomerase RNA only exclusively localizes to the Cajal body and not in nucleoli. Without TCAB1, they end up in the nucleoli, as you might expect. So did RNP map on this? What can this tell us about this process? We probed telomerase and found lots of expected protein binding sites with a bunch of new ones, including the region where this TCAB1 protein binds. And there's a atomic resolution structure of this already, so we can know and go in and map, like, yes, our approach is actually mapping exactly where these lysine contacts are being made with TCAB1. And what was really cool, so ultimately we have these cells that do or do not express TCAB1. We can do RNP map in both of those cell types. Telomerase RNA is about 450 nucleotides long. There was 7 changes that were actually significant across the whole RNA. And 4 of those, so most of them, were right in the binding platform for this TCAB1 protein. So RNP map is essentially giving nucleotide resolution information on individual protein binding events, which is good. Further, we could assign the sort of disappearance of a single cross-linking event to TCAB1. Sorry for how busy this slide is, but ultimately here we see a disappearance of this binding event when we knock out TCAB1. And we saw the same exact pattern in the TCAB1 binding site of another Cajal body localized RNA, even though their sequences are very different. This site 1 isn't binding TCAB1, which is why you don't see a change and you don't see a cross-link to begin with. Okay. So this was sort of the first inkling that indeed these profiles are very sensitive to single protein changes, and that these complex profiles could be shared across RNAs, even when they have divergent sequences. So we wanted to extend these findings to a model with a few more testable sequences. So again, it didn't turn out to, it's not my favorite RNA, but I've tested it a lot. We turn to the family of spliceosomal uRNAs, which includes this U1. So ultimately, non-coding RNA members of the major and minor spliceosome are all stably bound by this same set of proteins called the SM complex. It's necessary for their assembly. And so I had a recent rotation student look at RNP map profiles for 2 RNAs each from the major and the minor spliceosomes. You can see that the RNAs themselves are very different structures from one another, but they all have this SM core that is being bound by this protein complex. But you can see that the color is representing how similar the sequences are to one another, and they're not even identical in sequence, even though they're binding to the same complex. So the question was, can we actually see the same profile from an RNP map experiment in these different RNAs that have different structures? And this is preliminary results, but I was excited to find that, yes, I think there is profiles that you could probably even, by eye, I felt it was easy to actually pull this out. I did k-means clustering on this too, and it did separate it out into what my eyes told me too, so that's cool. But yeah, 3 out of 4 of these uRNAs, RNP map profiles, look similar at the SM protein site. So U1, U11, and U12 all have this 3-peak pattern, and the scale in which these peaks are also very close as well. While U2 seemed like it had this much more highly bound region preceding sequence. And if we just zoom back out and put these profiles onto the RNA structure, there's a crude way of doing this, but it pops out pretty easily why there might be a very different interaction between U2 and the SM complex. It's the only one that has this sort of extended linker. Not only that, the linker region immediately upstream of it is highly modified. It's one of the most highly modified spliceosomal RNAs. Whereas all these other ones, right, they still have this pattern of here's the binding site, and flanked on either side is a strong RNA secondary structure. So that was pretty heartening. Based on, again, preliminary results, I'm more confident now that we will be able to pull out meaningful profiles from transcriptome-wide data sets, building out a new encyclopedia of RNP network elements. I don't have that for you yet. That might have been exciting, but lastly, I want to talk about some more preliminary data, trying to adapt MAP to actually screen for small molecule binding to RNAs inside cells. So here's another area you can do a lot of simulation and modeling to try and predict ligands for protein surfaces, again, because we have lots of data on that. But we don't really have enough information about structure or ligand interactions to do the same thing for RNA. So I'm interested, an experimentalist, in coming up with methods that would allow us to screen inside live cells and learn more about what these interactions look like directly. So maybe we'll be able to model them and predict them going forward. And this is not a revolutionary concept. There's lots of people who are doing things like this. I want to employ these functionalized libraries. These have been used for proteins before and for RNA on a smaller scale. But I want to link it to a MAP readout where we can get nucleotide resolution information on ligand binding. And so, like I said, a small group of people have already used photoreactive and click-containing molecules for this, and ultimately get lists of RNA targets by biotin streptavidin pull down. We're kind of doing this too in a collaboration with Amanda Garner here. And there's a number of issues with this approach, just not as straightforward as you might think. So can we make an easier one? And I thought, because I only have stupid ideas, I figured we already have a pretty good enrichment method and mapping method for cross-linked RNPs. So why don't we just convert our ligand binding information into an RNA-protein crosslink? Then we could just use RNP-map as the readout and get both what is being targeted by this small molecule we're testing and what nucleotide is actually being bound by this small molecule. We started with an approach where we pre-installed a click handle into an RNA that would resemble the product of a screen, and then put proteins on it. And this is from a number, so undergrads and rotation students in the past have done this. I have a rotation student who's doing this again now, who was very excited to be pointed out. And multiple protein tags gave us near nucleotide resolution of where that binding site, proof of principle binding site is. And so now we're working on applying a test case inside cells for known ligand RNA pairs. And so if successful, this would be a new and straightforward way to screen functionalized libraries against RNAs inside cells. And so, okay, I've told you about multiple ways to find useful motifs in RNAs, mapping structure, protein binding, and ligand binding. And again, ultimately, the laboratory is focused on understanding how these RNA assemblies drive biology and disease, and how we might target them to control those processes. I don't have time to tell you about some of the biology we study in the lab, but we couple all this chemical probing to lots of different phenotypic assays and human cell culture, largely in models of lung cancer. And so with that, I'll acknowledge sources of funding over the years, the collaborators I've had. You'll notice like I'm not bolding anybody that's currently in the lab because I didn't show any of their cool work. But some of the people who did some of the work that I did show are highlighted here. And then with that, I will take any questions.
Thank you for your nice talk. I have two questions. The first question is, I think we can, I'm not sure how you calculate the energy level of the complicated RNA structures. I'm just curious if you are using a kind of Hamiltonian operator or quantum chemistry principles to calculate the energy level, because I saw some researchers are using that concepts when calculating the energy level of the high C data. And my second question is, I assume you have motivation to really see the RNA-protein complex structure, but I heard when using cryo-EM or something like that, it is very difficult and even unsolved problems to really visualize the structure of the RNP. So I'm just curious your perspective about this problem.
So I remember your second question, your perspective of how hard it is to get like cryo-EM structures of RNP complexes. For sure, like my goal would be to create new sets of data, both by our phenotypic assays, but as well as finding these motifs to narrow down these small functional regions that may behave better in these atomic resolution. I think I see those kind of atomic resolution mapping approaches for this kind of thing as the like last step, and like really, really knowing exactly what some of these things look like, represented by one part of one ensemble, ideally in one type of system. That give any kind of meaningful perspective. But yeah, I think though, because we can, if we're thinking about in general, trying to understand mechanisms enough so that we can perturb them to say, have some kind of therapeutic intervention, we don't actually need to know the atomic level resolution of some of these things most of the time. However, if we want to have information that we'll be able to then just pick a target, model it, and then come up with a drug very rapidly, that's what we'll need that kind of information for. So yeah, that's my off the top of my head perspective on that. And then I don't remember at all what you're something about energy levels.
Yeah. Actually, for example, when calculating the energy level of the benzene molecules, we can think of the Hickel approximation. And I'm just curious if you are also using the principles. We're just using simple calculation for calculating the energy level of the complicated structural RNAs.
Oh, I'm definitely using very simple method, and anything that's already been published 10 years. No, well, I'm being funny here. I don't think I can answer your question potentially as deeply as you might want it to be answered, is what I'll say, based on, again, the level of training that I've had. I'm still thinking of, because I come at it from what can we actually measure inside a system, and can we connect what those measurements mean or what we're seeing to what is actually physically going on. Yeah, I don't know. There might be a better way to restate your question that I could answer it in a way that you would want it to be, other than just being self-deprecating, but this is my personality, so.
Yeah, thank you.
I had a question about how the shape reactivity is doing the reactivity. When I was looking at the diagrams, I would have just kind of intuitively expected more symmetry, where you've got, your spoke, and it looked like just one side was having a ton of reactivity, the other side wasn't. They both looked just as open, where I'm thinking from like a chemistry perspective, I would think it'd be able to come in equally to both sides and you'd see symmetrical peaks. So I'm just curious how the shape reactivity is being measured / calculated, because I'm not understanding looking at it.
So when you're talking about shape reactivity and sides, which sort of, is there a slide I can go to?
Yeah, I think just any of the ones with the shape plot kind of earlier on.
Blah, blah, blah. Back in the beginning.
It's probably more of a biochemistry question.
No, I love this. So that's why I want to know exactly what you're talking about so I can give a... So do you want to look at it on a structure?
Yeah. Like on the structure.
Okay, let's go to U1 then.
Yeah, like this. So like, for example, on the bottom, it's coming up really red on the right side, but the left side, it's gray and not as reactive.
Oh,
so,
this guy?
Yeah. So
these are gray. So that's a caveat of doing the experiment. I can absolutely answer this fully. So the way in which we do this experiment, right? You react RNA, and then we could sequence everything, but that would be expensive. So instead, if we want to go after a specific target, we actually just use primers to amplify them from the pool. But if you use a primer to amplify a target, this will mean that you don't get reactivity information for where those primers bound. And so actually thinking about, remember, it's predicting this extra helix here. I could say that, oh, well, normally it's bound to a protein and it's missing this when we're doing this extracted from cells. That's why we're seeing this over-prediction of a thing. But additionally, it could just be we're missing the information from here. Because if you look at the other side of this potential helix, it's actually very reactive. So to me, even just looking at that, it's not even clear to me that this would be forming. Because there's no added shape information here, it's just going to favor making that because it has no inkling about if there's any level of reactivity on that part because the primer was shadowing it. I waffled between citing, I have a new paper where we have a different procedure that allows us to essentially get full reactivity information for these sort of smaller RNAs. And indeed, right, like there's actually a lot more caveats than when people present this kind of data. At least for one structure we tried to solve, it was giving us really weird results. It didn't look right. And all it took was actually getting information for, really was just like one nucleotide. If we knew the state of whether that was actually flexible or not, it would bias the model toward what was correct. So yeah, that's an important consideration.
Cool, thank you.
Great talk, Chase. For a lot of these methods, it looks like where you're going with it is scale in terms of finding transcriptome-wide drug screening, so on. I really like your ability of the OOPS to reduce this noise in the assay. And I'm curious if you could just sort of let us know where you're headed in terms of where's the next like source of noise and how are you thinking about trying to make that cleaner signal? And, you know, what's sort of the next piece to go after?
Yeah, so there's two sides to this. One, I could use help from probably people in this audience because cleaning up the data on the after collection side, I think there are ways to think about modeling what is an actual RNP map signal. What does it mean, right? So right now it's just modeling like, oh, it's the nearest lysine is 15 angstroms away or less. But I could think about modeling it in some of these things probably have an increased chance to cause a cross-link if it's near multiple lysines. So there's different ways in which we can think about, based on the data that we have, what does a reactivity at each nucleotide mean? Is it lysine proximity alone? Is it in a sphere? And actually, if you're thinking about the lysines that are in those structures, how much flexibility are in those lysines? This would take a lot of different modeling of that. So that I think is the after collection of data way I'm thinking about this. We can probably transform, because really I'd like to come up with something equivalent to how we make pseudo-free energy measurements with shape with an RNP map thing to say like, oh, these are actually specific distance measurements that we're making. But the other thing on the experimental side, I think we can enrich more. There's a way, because definitely I found that doing these OOPS experiments, you're definitely pulling along some other RNAs into that interface that are not because of a cross-linking event. So there's other ways to enrich where we're essentially labeling the N-terminal ends of, once we've cleaved off most of the proteins and then put them onto another column that will wash away anything that's not actually a protein RNA cross-link after that. So I think that might clean it up even more. But I mean, based on the ROC curve, we're pretty close to being pretty good, at least for predicting things that are within a certain region. So there's definitely more room for improvement. Yeah.
Do you see batch-to-batch variation? And also, can you do NovaSeq scale sequencing, or is this sequencing with these modifications? Do you require sort of a specialized sequencing protocol?
No. Definitely, it's nice to have more replicates of data. I think there are definitely RNA targets that are worse, but I think that has more to do with where you pick to make the amplicons. I actually think changing to a transcriptome-wide approach with random primers used for the RT step will actually make all those problems wash out. But definitely we can get NovaSeq level data for this, and it improves our ability to see more and more of those correlation events. But I mean, to get a binding site footprinting profile, every region needs to be covered by at least a range of thousands of reads. And then for the correlation data, maybe with OOPS now, maybe tens of thousands to start getting useful information. So yeah, that's why definitely thinking on the order of moving up in scale, that we get more information, and actually we can then use that to figure out what does actually a crosslink mean.
Chase, nice talk. Again, in this same slide, well, you have one on lot of reads piled up, and then modification, the sequence modification tell you the sites that are labeled. How long are those reads? Do you typically have multiple modified base per read?
Yeah, so I think it's in one of those images, but the normal length of a read, just based on how many amplicons or how big we make the amplicons, is about in the 450 range.
To cover that 160, oh, plenty to cover it.
Right.
But do you directly observe like a pair of modifications that correspond to the loops or co-occurrence of the jointly open sites?
Oh, you mean for structure probing?
That for your shape.
Yeah. So shape reagents don't do that as well as other structure probing reagents like DMS. And it's, I think it's just because of the nature of where they're sort of reacting. You can react with, shape reagents react with a 2' hydroxyl, and that doesn't necessarily change or deform an existing structure, so you could get a correlated event. But DMS and other reagents hit hard enough that ultimately when you get a modification, you melt the structure in real time. And so now if you pull apart the structure, now this attached nucleotide is available to be reacted with. And then you can start to see direct duplex detection, essentially, because you'll see base pair correlations.
Right. So especially when you have a dirty track, you suspect they are visiting multiple conformations. You have a complex mixture. Maybe those reads, by the time you cluster the stack of reads, some cluster correspond to conformation 1, another cluster conformation 2, right?
Yeah, so my a colleague who postdoc in the same postdoc lab as me, Tony Musto, he's been out here before too. He's sort of trying to push the limits of how you can do that sort of clustering methods to get at different members of ensembles within RNA structures.
Right. And what if you couple this with a library of RNA input, each carry a different point mutation? It's essentially saturation mutagenesis. And you use NovaSeq, it can provide this many millions of reads anyway. So you get to observe clusters of the mutational effect, right?
Yeah, I think, so I feel like Rishu Das has these mutate and map methods, which is similar to that. Yeah. Where you sort of look for changes and then see like, oh, which of these things perturb each of these structures, and get a set of potential structures that way.
Right, because even for experimental evolution, people observe pairs of mutations have, say, comparable fitness as a way to see which are paired in the stem. Great, thank you.
Any other questions? If not, Chase, I have a very naive questions. So RNA population is definitely very diverse, very dynamic. I love the ideas of the chemical probing, but can we measure the variation of the secondary structures or the RNA protein interactions when we utilize this technology?
So,
like the heterogeneities of the RNA secondary structure.
So definitely you need some kind of clustering approach to separate out what reads look more like other reads. And people are definitely doing that with structure probing data. I don't know if the RNP mapping, because not every nucleotide is going to be bound by a protein, and especially not every protein RNA interaction site is going to require a lysine. So just the number of potential sites we can measure in one RNA is already, the fact that we get correlations at all that are meaningful is sort of, I think, probably indicative that when you get a protein complex forming, like it's there, that's meant to be there. It's a fully stable complex. But we won't get, I think, as many modifications as we need in a single read to actually be able to cluster them, meaning at least for RNA protein interactions. One way to cluster them I've started playing with is like looking at RNAs that are, but this is asking a specific other question, looking at RNAs that have different ends, right? So things that are tailed at different lengths, things have immature RNAs or some 3' degradation is happening. So we can pull that information out with sequencing and see like, oh, if this is not fully assembled, what does this look like? What proteins are bound to it when it's not in its active form? We can do things like that with the RNP mapping so far, which is an interesting thing. We're doing that with telomerase and looking at as it's trafficking through and being assembled, where do proteins come on, where do proteins come off? But in general, most of the stuff we looked at tends to form one thing. It has evolved to make one complex, at least in local region. So they look the same every time we probe them. But we can't really know that there isn't an ensemble of stuff within that data. Definitely fractionating out things we can see in the nucleus, this complex forms like this, and cytoplasm, it forms like that.
Thank you so much, Chase. Questions? Okay, thanks, Chase. Wonderful talk again.